It's really great to be here. This is my cousin that you all know. I think it's been a wonderful thing that he's organized. How many people here know their own genetics? Anything about, other than Ancestry? I know you're all Ancestry thing. A little bit. I think that will change very soon. And so you might want to learn some more about it. And so some speakers will ask you not to bring out your cell phones and text and so forth. But if you want, you look at pged.org. It's a organization my wife started for personal genetics education. And you'll find some fun things there, like what's on the next slide. And so we're going to talk about how you can learn about genetics and how you can share. The previous talk was a perfect introduction to that thing about giving rather than taking, or giving and taking. And so this is some of the stuff on the Personal Genetics Education Project. They do things like work with Hollywood and their TVs and films. They have a little map where you can they ask you questions, very simple questions, 5 questions, and if you once you get them all right, and everybody does, then you get to put a pin where you live or where you are or whatever you want. And for example, here's one where I pinned myself just before coming here. And so the little mark there is where you are. And there are tests on Alzheimer's, genetics, and alcohol, and genetic discrimination, and Ebola, and all kinds of fun topics that you should know about. So we have a strange family here. And in this project we call the Personal Genome Project, which is a project all about sharing. By the way, I'm going to hope that at the end of this we might have a little audience participation. So if you have any questions at all on any of the things I'm talking about, save them up, because I'll end a little early. But anyway, this Personal Genome Project started about a decade ago, and here we have it looking a little bit like mugshots, the first 10 volunteers. I was asked by the board that reviews the ethics to participate in the beginning, to eat my own dog food, as it said. And so I'm sometimes called guinea pig number 1, or PGP number 1. And you'll see some of that process in a moment. And then fast forward a few years, and the family is a little bit bigger. Every year we have a conference, kind of like this one, where we get together not just scientists, but patients and people that are volunteering. And what they're volunteering is really quite extraordinary. They're volunteering to have all of their DNA, everything we know about their environments and their traits, medical and non-medical traits. You'll see a few examples in a moment, such that scientists and anybody with an idea can test them out using these, what's now called precision medicine or personalized medicine. They can get a big data set off the internet for each individual and test ideas out. And then this is a year later. Now it's expanded from Boston to Boston and Toronto, and a year after that. It's now in 5 parts of the world, and about a dozen are waiting for approval in their respective countries. So you get, it's kind of like Wikipedia. It's distributed. It's volunteer, and each country has its own recruiting, its own fundraising, and so forth. And so, and there's one now in New York. In fact, the headquarters are now in New York. Some of you may have seen Skip Gates earlier here in the same room, and he is certainly a figure in the ancestry field. And I had the pleasure of being with him on Faces of America. This is a shot from that documentary. And one of the things that both he and I worry about, or think about, enjoy, is mapping migrations of people, and Spencer Wells, you heard earlier as well. And some of the really ancient things go all the way back 500,000 years to the Neanderthals, and maybe since then when we've interbred with those and the Denisovans. And so that's Skip with Stephen Colbert. Neither of them being funny, both of them having a good time looking through ancestry records of the Colbert family. And then a few months later, not on Faces of America, but on the Colbert Report, here's my book that Stephen is hawking for me, which happened to have also been encoded into DNA. just to keep the DNA theme. And so there's 20 million copies of my book in his hands. We made, in the end, 70 billion copies of the book, just for kicks. And then, of course, his first temptation is to eat the 20 million copies of my book. And I tried to stop him from doing that. So what good is this to you, all this DNA, you know, whether it's in what is the point? And what is the point of sharing of yourself? What is the point of bringing your DNA, your medical records and so forth in a shareable form so that physicians and scientists around the world can do something about it? Here's some examples, some kind of family examples. These are the Beery twins who I've met. I'm not their physician or anything, but they had, their family was very dedicated to figuring out what was wrong with them. They were misdiagnosed with cerebral palsy when they were young. They were very lethargic. They could barely walk or lift their arms. They couldn't really make it through school. And eventually, out of desperation, the physicians involved sequenced their genome, not just a few pieces of it, but all the genes, and found out that it was due to one gene in bold there, SPR. Doesn't really matter what it stands for, but the result was that they take these two oral supplements, which you can find in your average drugstore without prescription, which are in the serotonin and dopamine pathways in the brain, and now they are completely normal kids, you know, when I last talked to them, they were just as happy and energetic as any teenagers you could find. Maybe more happy and energetic than your average teenager. Here's another person. John Lauerman happened to be in our Personal Genome Project, and here he is getting his blood drawn by one of the physicians in our project, Joe Thakuria, and he sort of initially presented himself as healthy, but then when we found on further inspection, he had scotomas, which are dot spots in your eye, in your visual field, and leg pain. Leg pain so severe that they had done a little bit of genetics. They had looked at one gene. Now that's a mistake, because if you just guess the gene, you could guess wrong. There are over 30,000 genes, and you guess one. Anyway, it turned out that when we did all the genes, he had a JAK2 mutation. It was quite striking, and it was in his blood, and not in every single tissue of his body. So that's another interesting lesson, is we are not homogeneous. We are a mosaic of different genomes, but we're mostly what we inherited from our mother and father. In any case, so he's taking aspirin for the rest of his life, and the scotomas and the leg pains have gone away. And he wrote about this in Bloomberg. So far, it's just taking different things as part of your oral ingestion, and this is not very painful. But the third example, I think, involves a great deal more sacrifice, but it has a definite upside. So in this case, hopefully you've all heard of Angelina Jolie's story, which is that she looked at her genes and found BRCA1. It's probably one of the most famous human diseases, that gene that makes you susceptible to breast cancer. And so since her particular family history and genetic information indicated she had 87% of breast cancer and a slightly lower percent of ovarian cancer. And so what's often done is removing the tissues that are at risk before they develop even the least bit of cancer, because once you develop a little cancer, then it metastasizes and it's all over the body. But if you take the tissue out before it's developed cancer, you can completely avoid the risk. And so there are millions of women and some men who have taken this test. And it doesn't always mean that you have to have it in your family. Essentially, all of us are at risk for everything, and you don't know that you're not at risk until you take a look at your genome. Now, you might be saying at this point, I can't afford that. That's ridiculous. I heard that it was $3 billion to get your genome sequenced. And it was indeed, when we did the first genome, it was actually a pretty poor quality genome that we did for $3 billion. And I'm happy to report that today we can do a much, much higher quality genome for about $1,000. And hopefully within a year or two, we'll be down around $100 and it'll keep dropping. It's dropping faster than the incredible rate at which electronics has been dropping. This is an exponential with factors of 10. So even if it had been going even as fast as computers, it would have taken 6 decades to drop down to affordable, but it only took about 6 years. So I'm not going to go through all the technologies. I mean, our lab was heavily involved, and these are some of the companies involved. But I'm going to give you a little insight into what might be the next thing, and I'm pretty excited about this. These are nanopore sequencers. The sequencer that did most of the data that represents this slide, most of the sequencing in the world, maybe 100,000 people so far out of 7 billion, was about this, maybe 5 times the size of this podium. It would be measured in, tons in the original sequencers, some of them. Now you have something that fits in the palm of your hand, hooks up to your computer through a USB port, and so we will soon have, just like on your cell phone, some people have 2 cameras on their cell phone. You'll have 2 cameras and a genome sequencer that will, you say, but my genome doesn't change from day to day, right? Well, but your environment does. So if you walk into a room full of H1N1, you might want to know that, and your little handheld cell phone sequencer will tweet to all your friends that you've been exposed to H1N1 or Ebola, whatever. So back to this project, which you can all participate in. Our goal is 7 billion family members. All of you are invited. And so you can go to the website personalgenomes.org. It's a nonprofit. And it's the world's only open access data set. So Congress has mandated that scientific data in general be open access, but nothing in human genetics is as open access as this. And it's not just about your genome and your microbiome, the things that put you at risk in your environment, but we take data in your brain, so magnetic resonance imaging, MRI. This is for my brain, since I'm guinea pig number 1. I want to make sure. These are virtual slices, not actual slices on my brain, in case you were wondering why I was having trouble speaking here. And then these are cells that have been taken out of my arm. They're just a simple skin biopsy that doesn't hurt, and it just doesn't require any stitches, just a little bandage. And then they can be turned into stem cells and then into these various differentiated tissues, including brain components. These families, so-called trios, mother, father, and child, are now part of the National Institute of Standard Technology and Food and Drug Administration standards. So some of our participants in this project are now immortalized the way that the meter stick and the kilogram weight are immortalized as standards for the entire world. So other fun things you can do with DNA. DNA is not your destiny. You may know that it's not your destiny because the environment is a big component. But it's changed radically in the last couple of years, because not only can you change your environment and therefore change your destiny, but you can actually change your genome. You can change your DNA. And here's an example from a company called Sangamo, and there are 2,000 clinical trials right now, all of them looking very promising, where you can genetically engineer cells in your body, in this case to make them so that your cells that normally would be infected by HIV are resistant. They don't have the virus receptor anymore because you genetically changed it. That's pretty radical. These are patients that have full-blown AIDS. They could even have drug-resistant AIDS. We don't have a vaccine or drug that's perfect, but we can make it so the virus can't get into the body. Now, this is another kind of family portrait. Here they've arranged themselves by size, and you show the kind of the typical bell curve that they like to teach in school. So you get a lot of people in the middle size, and then rarer and rarer as you go to the end. And they didn't even show the real extremes, which are out there, which are less than 5 feet, more than 6'5". And you can do genetics on this. And one of the biggest cohort, that is to say, a group of people for which medical genetics is done, this is one of the biggest ones. It's about 250,000 when this slide was done. It's bigger now. And it looks very confusing. You know, we've got thousands of different DNA variants that affect height by tiny amounts, and you say, well, there's nothing we can do about that. There's nothing we can do diagnostically or therapeutically. But it turns out that if you look way out on those extremes, both ends, very short and very tall, it becomes much simpler, and it tends to be growth hormone and the receptor for the growth hormone. And you get this dynamic range of people as small as 2 kilograms and as much as 600 plus kilograms. And that insight into that one gene, human growth hormone, results in therapies that go beyond the individual that might be deficient in that might have a mutation in the human growth hormone gene, to all these different diseases. Now, I showed you that bell curve of people standing sorted by height. Here's another bell curve. Height isn't really a disease. There's various human growth hormone treatments, but but this is a common disease. This will kill almost everybody in the developed nations like the United States. 90% of us will die from this disease. Some people don't call it a disease, but it's all age-related diseases. The aging process itself is programmed into our genome, and that's why it is that mice die after about 2.5 years, and bowhead whales die after about 200 years. They're both mammals, similar body temperatures, but this is programmed. But there are some people who sort of escape a lot of the damage. The take-home lesson for the slide, by the way, is not to drink and smoke and eat sugar to excess, but actually that these people manage to get fairly old despite their environment, not because of it. They have, Jeanne Clément was riding her bicycle, I think, until she was 110, and she smoked for a century, stopped not because she had a lung disease, but because it was less socially acceptable 2 centuries after she started. Now, I mentioned the cells that we get from the Versatile Genome Project, and these can be reprogrammed, and LOAD is an abbreviation of late onset Alzheimer's disease, and you can edit these cells so that they can become more or less prone to Alzheimer's based on genetics. And you can also get cells from patients and reprogram them into neurons. And when you do that, you learn quite a bit about this. And Rudy Tanzi gave a lovely lecture on that earlier today. And we can build we are in the process of building the parts of the brain that are most sensitive to Alzheimer's. Some of the earliest things that happen are in the hippocampus. And so we're building more and more faithful models of this hippocampus in the human brain. And again, these are cells from my body, but they could have been from anybody's, which have been reprogrammed. And I'm gonna hopefully been provocative enough I've encouraged some questions. And I'm gonna urge you again to go to pged.org, go to the map part where you can ask questions and answer questions and pin yourself on the site, because there are not very many pins right now in Queens or wherever you come from. So I'm going to open it up for questions right now. Any topic from the entire meeting if you want, but ideally from mine. Yes?
So using this that I just heard, can I somehow reverse aging if I find out? Can I do that now?
Okay, so the question for everybody, maybe didn't hear it, is can we reverse aging today? And the answer is there's no FDA-approved reversal of aging, but we are making tremendous progress in animals, and so there are some combinations of genetic tricks that can get certain animals, like nematodes, to live 5 times as long as they normally live, and some of these are now have moved into mice, and you can get mice to live twice as long as normally, and then we're in my group, as we move them out of mice, we're doing dogs next because people care deeply about their pets, and the FDA approval process is much faster for dogs than it is for people for a variety of reasons. And we think that that we get feedback on any unintended consequences is the main thing you want to avoid. You don't want your drug to fail, your therapy to fail. And so I think people pay more attention to their dog's welfare and mood and so forth than they do to their mice. So, if your dog is having a bad day, you typically know it. But if your mouse is having a bad day, you typically don't. But there's tremendous progress. I mean, we hope to be doing clinical trials in dogs this next year, and then humans will be using some of these new DNA manipulation procedures in 2016. Thank you. Any other volunteers? No, sorry. Or questions, rather. You had one. Right. Yeah. Great question. The question is specifically on genetics of aging. Are we looking for duration or quality? And what I could say from my colleagues and myself, I can't say for the entire field, we're focusing on aging reversal rather than on longevity for two reasons. One is very practical scientifically, which is to do a longevity assay on an animal even remotely similar to humans, like a mouse. That's a 5-year project, which is a long time for a graduate student's career. And the other reason is, and so with aging reversal, there are procedures where you can see within a few weeks whether the cognitive function is improved, the reaction times, they have grip tests and things like that. And so there are some examples. The most dramatic one is something, I'm going to sling some jargon here, heterochronic parabiosis, which just means that they sew together a young and an old mouse. I don't recommend doing this with your babies or anything, but yeah, you've heard of this, yeah. So that we're just beginning to mine the factors in that blood system exchange, but that's aging reversal. Another example, not a particularly good one, but we can take my cells from the skin, which are 60 years old by a variety of criteria. You can molecularly characterize the methylation on the DNA, say, and you can draw a straight line through everybody in this room, and you can predict your age within 2 years by our epigenetic modification, 2-year precision, and mine comes out to about my age with about 2 years. You can now reprogram those to stem cells and back to fibroblasts, back to skin cells, and they now look like they're a newborn's cells. Now, that's not appropriate for my entire body, but it's a proof of concept we can do it for one of my cell types. So there's 2 examples right off the bat that are working in mammals or human cells. Good question. Yes?
To the DNA about our destiny, so which group of genetic diseases basically have a leg up in the research world to get cured?
Yeah. So the question is, which diseases are ahead, in a certain sense, in getting cures? Is this a political funding issue, so forth? I think to some extent, the big breakthroughs recently have been enabling technologies. So in a way, they were democratization. They weren't political. They floated all the boats. So we recently brought down the cost of reading genomes, so now that's why Rudy Tanzi can say that he, glibly say that he did 3,200 human genomes, like that, not $3 billion each. That floated every disease happened. And then now the ability, that's to read it, and then the ability to write it to do gene therapy has jumped forward in the last few years. One word that you'll hear more and more, I didn't put in my slides, but just give you some jargon to take home, is CRISPR. So it's like, it's like in The Graduate, I have one word for you, my son, plastics. Well, the word I have for you is CRISPR, okay? Yeah, okay? But also in terms of diseases, I think they are prioritized roughly based on their prevalence of the population. And that's why I picked the most prevalent, the one that kills 90% of us and predisposes us to many of the other ones. Now, the genetic diseases already have some solutions. There's genetic counseling. There's PGD IVF, which is prenatal genetic diagnosis combined with in vitro fertilization. So these things are already having an impact. For example, Tay-Sachs is a very devastating disease, terrible for not just for the child but the family, and that's almost completely eliminated in the populations that have adopted genetic counseling. Yeah. Right. Right. So let me rephrase the question for everybody who didn't hear it, and to make sure that I understood it before I start answering it. So you're concerned about particular disease and classes of diseases. And you're asking, what's the quality of life? When will we be ready to fix the genes? Is that close enough? Is that, or let's start with that, and then you can correct me if I don't answer it. There are some diseases that are very hard to fix when you're late in life because you've gone through a developmental process and it's become irreversible essentially, right? But there are many, many others that seem irreversible that can be reversed, a surprising number. And I'm not going to snow you with numbers or something like that, but I think each year we discover more and more things that are. An example of something where you have to hit the development right and for which it's a terrific success story, so I'd like to tell it, is just as a preface, when you do these clinical trials, there's preclinical, which you do in animals, and then there's phase 1, 2, and 3, which you do in humans. And then there's the rest of us, phase 4, when it's open to everybody. Anyway, there's congenital blindness, which is a developmental problem, and you lose, and there are various ways that it can go wrong in your retina. There are gene therapies that have been applied already in phase 1 clinical trials, which is the first one. We were basically just looking to make sure it's not toxic. They rounded up, it's a fairly rare disease, they rounded up everybody who had the disease that was willing to participate in the trial, cured essentially all of them, and now there's nobody left to do phase 2 clinical trials. So I'm glad everybody gets that. So it's a wonderful problem to have, okay? Now I'm not saying, but it's critical, the thing that's relevant to this developmental thing, it's critical that you do it while they're young, because otherwise if you try to reverse it later, they will be able to see, the photons will come in and will register in their brain, but they can't really see faces despite a lot of therapy. So it's very much case by case. And it changes with time as we do research. I'm sorry? Do we take genetic samples? Yes, so the question, I think you're talking about the Personal Genome Project. We take samples wherever there's a conference. So the next conference will be in Vienna. But it moves around. We've done them in Mountain View in New York and so forth. And you can encourage, if you get together some people, you can encourage it to happen in a particular town. Yeah. We have done in the past saliva, which can be done through the mail, but we're not doing that so much right this moment. Yeah.
One more question, one final question.
Yep. Right. So the question for everybody is a particular type of macular degeneration in the retina, and that is one of the best studied genetic diseases for which there are known causes I'm not saying we know every cause, and that's true for almost every disease. We don't know every cause of every disease, or of any disease. But I think there will be gene therapies for that very soon. The retina is a particularly nice place to do gene therapy. That's why this blindness was one of the first that was done, because it's really a very accessible in an outpatient clinic to just, it doesn't sound pleasant, but it's better than a lot of other treatments to inject a gene therapy into the retina.
Okay.
There is studies on both dry and wet. There are many genes involved in both.
Okay.
So thank you. You've been a great crowd.
Professor George Church, everybody.